Pleasure for me to introduce today's seminar speaker for the Frederick National Laboratory for Cancer Research Seminar Series, High Fidelity, Dr. Marc Ferrer. After completing a bachelor's degree in organic chemistry at the University of Barcelona, Marc relocated to the Twin Cities to earn a PhD degree in chemistry at the University of Minnesota. During his postdoctoral fellowship at Harvard, Marc developed small molecules that inhibited the viral entry process of HIV. Subsequently, Marc spent almost 11 years at Merck, where he served as the Director of Assay Development for lead and target identification, leading a group of 20 scientists to develop and miniaturize biochemical and cell-based assays for lead identification within Merck drug discovery programs. Marc is currently the Director of the Intramural 3D Tissue Bioprinting Laboratory at the National Center for Advancing Translational Sciences, or NCATS. This laboratory is a multidisciplinary group that produces, validates, and uses 3D biofabricated tissues for disease modeling and drug discovery. Created as the result of NCATS's effort to develop and use complex 3D cellular models for drug discovery, this laboratory is a core resource for collaborating with the scientific community to biofabricate functional human tissues in multi-well plate format, which is also known as tissues in a well, using relevant human primary or induced pluripotent stem cell-derived cells to enable disease modeling and predictive toxicology and efficacy in preclinical drug testing. In this collaborative model, the laboratory provides biofabrication and drug screening expertise, while extramural researchers provide disease-relevant and 3D modeling expertise. As you can imagine, success with this bleeding-edge work is critically dependent on standards, reference materials, and reproducibility, which is the major focus of the seminar series. To unite researchers throughout the world in developing and adopting standards and reference materials in this rapidly evolving field, Marc established a 3D tumor model standards working group 4 years ago, including stakeholders in government, academia, and industry who are focusing on the standardization and advancement of 3D in vitro model systems that correlate with patient responses. With that, I will welcome Marc to begin his presentation, and just a friendly reminder to everyone to please mute your audio connections unless you are speaking. Welcome, Marc.
Thanks so much, Nathan, for the kind introduction, and thank you so much for inviting me to this series, which I believe are very important. I sort of like your name of your program, your Star Trek Standards Reference and Training Research Committee. I love the acronym, but it is an important issue, the standardization of these 3D models that we're trying to develop. And what I thought I would do today is sort of give a little bit of introduction as how we got into 3D tissue models, and then give you a couple of examples of the work that we've been doing to develop these models and standardize these models and use these models for drug screening. And one of the examples is going to be on skin tissue and a model of atopic dermatitis, eczema. And the other one is I'm going to be showing some of the work that we've done in the last year, developing long tissue models for COVID-19 discovery of therapeutics. So can you see my slides? Let's see if I can be a bit. Can you see it?
Yeah, that's perfect now, Marc.
Okay, let me minimize this. Perfect. So I don't think I have to spend too much time explaining how we got into 3D models. And the idea is that 90% of the drugs entering clinical trials fail, and mostly because of lack of efficacy, but also because unexpected toxicity in humans. And I think it's the reality in the field that the reason for that is that the in vitro models that we use, cellular models that we use, and the animal models that we use are really not that predictive of what or whether a drug will be effective in humans. So therefore, we really need in vitro models that are more predictive or more accurately predict the effect of drugs in clinic. And therefore, there's been a lot of interest in developing what some people call complex in vitro models, some people call microphysiological systems, some people call 3D organotypic models. The reality is that at some point we probably need to agree on a sort of a nomenclature, and that's part of the standardization. But I think if we, in general, we call these complex in vitro models, there is a range of models that scientists have been developed for decades now. And there's been a lot of work with spheroids, especially tumor spheroids, essentially assembly of cancer cells into spheres. And then there's been a lot of research also been done on organoids, which in my mind are really more generated from iPS cells or progenitor cells through a process of differentiation. And some of these organoids really have sort of tissue-like kind of structures in them, which the spheroids are sort of essentially cell aggregates. And then what I'll talk today is a lot of the aspect of bioengineered tissues or biofabricated tissues, which is an area that perhaps has not been developed so much for drug screening. Has been developed a lot for regenerative medicine, but not so much for drug screening. And then there's the whole aspect of organ on a chip and microfluidic systems. And as you go from left to right, indeed, the physiological complexity of these models sort of increases, but that comes with the price, especially for disease modeling and drug screening, that your high throughput compatibility goes down. So if you are in the business of drug development, you really have to sort of think as to what is the best model that you should use for whatever stage in the drug discovery process you're interested in. And regardless, the reality is that regardless of any of these 3D models or complex in vitro models that you're using or you decide to use, there has to be a process of validation. And I think the field at this point is at a stage where technically you can make many of these complex models, but I think what we need is a sort of a more rigorous standardization of what's in each model. So there's what some people call the scientific validation in terms of what are the cells in these tissues, what's the morphology of these tissues, what's the physiology of these tissues, and really how close are you to a native-like tissue. And that, I mean, you have to do that for let's call it a normal tissue, but of course, many of us are interested in developing disease models so we can find treatments. And then what you need to do is really, how do you benchmark these 3D models that you're making to assess, first of all, how close are they to a clinical state of the tissue? Can you use clinical biomarkers and establish that your model really reproduce some of the relevant clinical biomarkers of a disease? And then really, then within a model, establish like, okay, so diseases are very complex. They normally involve more than one tissue, and there's both sort of parenchymal, systemic interactions between cells and organs. So the question is, what are you capturing? What in your model? It's very difficult to capture a whole disease end to end, and the different aspects of the disease. So that some of the organ on a chips are trying to capture that complexity. But really, for spheroids, organoids, and even biofabricated tissues, what you're trying to do is really capture some aspect of the disease. And knowing what is that you're capturing, it's called the domain of validity of a cellular model. And that's that's a very important aspect of establishing the scientific validation of your models. So you convince yourself that you have a biomimetic sort of 3D tissue, then you have to worry if you want to use it for drug screening or therapeutic screening. You need to worry about the technical robustness of the tissue. Can you make it? In our case, we're interested in making tissues in a multi-well plate format, so we can use it with our automation capabilities. So we really need to go through a robust process of establishing the reproducibility of making these tissues, both the normal and the diseases, and really sources of cells. Can you, does it work with different, if you choose to use primary cells, what batches of cells does it work for? If you change your lot of primary cells, is it going to reproduce what you get or not? If you use iPS-derived cells, if you use one clone versus another clone of iPS cells, are you going to get the same? How long does it take for a tissue to mature and get to the state that you're interested in testing your compounds? And once it gets to that state, how long does it stay? What is the lifetime of tissue? So this process requires a very, very rigorous sort of establishing, like whether it's like protein markers, whether it's genomics markers, transcriptomics. So you need to have a panel of assays that you use to QC and establish the relevance and robustness of these tissues as you manufacture them. And so you have nice tissues, it's robust, and they are robust, but then you're gonna use these tissues for screening. So at some point you need to develop an assay, a functional endpoint that is relevant to your disease of interest, and make it work in a 3D model. So a lot of us are used to doing high content screening and reporter assays. So there's another level of complexity when you go to these 3D models into the physics of some of these assays that make it really difficult. So thinking about how you're going to be developing, what technologies you're going to be using to measure a phenotype in your 3D models that is compatible with high throughput screening, that's a huge need as well. I mean, you can use histology to validate your tissues, but you won't be able to use histology at the moment for screening. So you have to think about that. And then finally, you need to establish some sort of pharmacological validation. So you have a disease and you need to prove that you are able to correct the disease phenotype with some sort of pharmacological intervention. And that's something that I'll show you a case, but it's an important step in the development of any assay, both 2D and 3D. And so we wrote a review on this topic with Jens Kalm and Sita Madulal at the FDA, and that was published a couple of years ago. And there's also a nice review by Jason Eckert and the GSK group on SLAS discovery last year that discusses a lot of these topics, I think, in a very relevant way. So we got into 3D cellular models a few years back, and we were doing tumor spheroids and doing screens with tumor spheroids, but then the opportunity came along to implement tissue bioprinting techniques to create 3D tissue models. And since then, we've been able to create this lab, and I think Nathan described what the goal is. The lab is really a multidisciplinary lab to biofabricate tissues in a multi-well plate format for disease modeling and predictive both safety and efficacy drug testing. And what we're doing now is create a portfolio of tissues, both normal and disease, that we can use for screening. And at NCAT, we also have a very sort of very good infrastructure in terms of high throughput screening, chemistry. So we're trying to leverage all these capabilities that we have in CAS to still really create sort of a good sort of ecosystem where we can pressure test these 3D models that we're developing in a real sort of drug development pipeline. When we got into 3D, there was, the assumption is like, just because these 3D models are sort of look more like tissues, the expectation was that drug responses would be more like in clinic, and the truth is that we need to prove that, right? So questions that we're often asked and we're trying to ask and answer in the work that we're doing is really quantify, or try to quantify a little bit better how much better are 3D tissue models than 2D models in predicting drug outcomes in clinic. Tissues are very complex. They have very different cell types. So questions like how much cellular complexity is needed in a tissue to model a disease or some part of the disease, and I'll show you some examples of this. And then, can we reproduce clinical biomarkers of diseases in these 3D tissues? That's a very important aspect. If you are trying to, if you're saying that these models are going to be more predictive, then the next question is like, can you reproduce any of the disease benchmarks or the disease features that you see in clinic in your tissue and correct them? And then, if you can solve the issue of technical reproducibility and get good statistics from well to well, can you then leverage these tissues by perhaps using cells from different sources, from different diseases, from different populations, like whether it's race, gender, whether it's populations with asthma, with hypertension, and can you, once you solve the issue of technical reproducibility, start sort of looking into your disease and see if any of these differences in patients make a difference in your tissues, and like sort of going into the personalized medicine. So with all these questions in mind, we thought that 3D tissue biofabrication would be a good technology to use, because biofabrication and tissue engineering techniques, including bioprinting, really lets you control what cells you include in your tissue. It lets you control the sort of layering of the cells in a particular morphology that sort of it's more native-like, and really include other physiological features that perhaps are difficult to include in organoids and spheroids, like vascularization, innervation, and really because our approach was to use either primary cells or iPS-derived cells, essentially mature cells, then the time to readiness of an assay is really shorter. So we were hoping that we could get more reproducibility and really be more compatible with a sort of, let's call it medium throughput HCS, a 96 well plate. So with that in mind, we set up sort of an effort in the lab to create bioprinting tissues, and was an effort that was initiated by Sita, who established a collaboration with Organovo. And just as a sort of bioprinting 101, conceptually, it's actually not that complicated. You have some sort of hydrogel that you mix with the cells, you load it into a syringe, you load it into a bioprinter, which has a computer-assisted design software that allows you to create some sort of pattern. And most of our tissues are really in a transwell plate format, as is shown here. And what the bioprinting, again, allows you is to have very good spatial control, very good reproducibility. And where we've seen in some of our tissues, it allows you to, again, miniaturize the tissues to 96 well plate, which for us is huge. This is just a couple of movies. You can have different ways of dispensing. In the lab, we have a couple of Regenue discovery bioprinters. So one is sort of an extrusion base, the other one is an inkjet base. So you can use all these capabilities within the bioprinter, depending on what your needs are in terms of making the tissues. We spent perhaps a couple of years learning really to create cellular patterns, and these are just images using fibroblasts expressing either red fluorescent protein or green fluorescent protein, and to show that we can actually extrude these cells with a hydrogel and create spatial patterns both in 2D and in 3D without sort of killing the cells, and in a way that the patterns remain in the well. So now I'm gonna sort of show the first example, and as I mentioned, we started working with skin tissues, and there was a lot of work already being done for decades, a lot of it being done in Europe, because in Europe you cannot really test compounds in animal sort of compounds for dermal use in animal models, so you have to by law use this skin tissue equivalence. So there was a lot of work already been done in Europe, and we sort of started with those protocols to create biofabricated skin tissues. And as you can see in this cartoon, skin can be a very complex tissue, but sort of going to the basics, it's really a dermis base of fibroblast and ECM, and on top of it there's the sort of the epidermis, mostly made of keratinocytes that once they are exposed to an air-liquid interface, the skin is exposed to air, they differentiate into different layers. So when we started working on skin, essentially the idea was to use a transwell setting to make a dermis with fibroblasts and ECMs, and then a sort of epidermis with stratified keratinocytes in the air-liquid interface. We thought that with all the work that had been done, that we were just going to reproduce it, and sort of part of what we wanted to do was really, again, not just making the skins, but making the skins in a reproducible way in a multi-well plate format, and being able to use these tissues for high throughput screening. So one of the issues with the protocols that existed was contraction. So our first sort of first years working with skin was to develop protocols that would prevent contraction, and that's what we published now a couple of years ago. It's a fully bioprinted skin tissue, full thickness with the dermis and epidermis in a transwell, multi-well plate format that didn't contract. And we actually spent a lot of time making sure that these tissues get good barrier function in the well, so without having to take the tissue out, put it in a front cell or some sort of manipulation. So we did sort of a lot of barrier function assays from the top, from the bottom. We made sure that the keratinocytes had the right sort of morphology. We showed that we could use it for cytotoxicity testing. And then we used technologies like optical coherence tomography to show, without having to do histology, really it's sort of a non-invasive, non-destructive technology to look at the structure of the tissue and show that in the well, the tissue had the right sort of morphology and layering. We developed also protocols to include vascularization into the tissues, and this was work that was done by MJ Song and Lucia Liu in the lab. So this is something that we've been very interested in, sort of a progressive approach in cellular complexity, and how we can use or develop modular and versatile protocols that allow us to, in sort of a reasonable amount of time, to increase the complexity of the tissue. So this was a protocol that we published last year, in which we were able to create a vascularized dermis. This is not perfused, but the vascularization, as shown here, has a lumen. So what we did essentially was to mix endothelial cells, parasites, and the fibroblasts, and create this vascularized pattern with angiogenesis in the dermis. And again, as part of the standardization of the tissues, we do a lot of histology with the relevant markers and a lot of 3D microscopy to really establish the biomimicry of these tissues. So what we've done for several years was to create this screening platform of 3D human skin tissues. And it ranges from just skin epidermis, and those you don't need a bioprinter to do. And those were protocols that were very well established for years. And then now we have a full thickness skin tissue that is bioprinted, biofabricated. And then we also have a vascularized full thickness skin tissue. And here I'm just showing the protocols of how we make it. And essentially, you put the keratinocytes on top of the transmetal insert, and then after a few days submerged, you go into the air liquid interface, and that stratifies into an epidermis looking tissue. And then you can choose to put either the fibroblast at the bottom, so you create a full thickness skin. And I mentioned that we are able to also create vascularized by mixing endothelial cell fibroblasts and parasites. So with that in hand and the validation, we decided to show that we can use these tissues to actually model a disease. And we started with atopic dermatitis of eczema, which is an immune disease. It's a very complex disease. And when we started sort of discussing how to develop a model, it was really, it's like what stages of eczema or atopic dermatitis do we want to sort of mimic? So it starts in the non-lesional with some sort of allergen that triggers some sort of immune response that goes all the way to the lymph node. And then they secrete Th2 cells that go back to the skin and secrete cytokines like IL-4 and IL-3. That's really what induces the barrier disruption, the itch, scratching, and that is the sort of farther downstream signaling, which is the chronic stage. So it was very difficult to really sort of model the whole sort of end-to-end atopic dermatitis disease. So we decided to focus on the acute stage of the disease, and essentially what we did was to use IL-4 to induce or try to induce atopic dermatitis phenotype in the different disease models that we had. So that's what Lucia did. So essentially what you do is you use your sort of normal skin protocol, and then once you go into the air liquid interface, you add IL-4. And then what we did was to sort of after day 21, you take your tissues, you do histology, and you ask yourself, do these tissues look like clinical samples of atopic dermatitis? And what I'm showing here on the right is the normal vascularized skin equivalent, nice sort of histology. This is the epidermis, this is the dermis with the expected markers of skin differentiation. And then what you see with the IL-4 is this hyperproliferation of the keratinocytes, and that some of these biomarkers disappear, which is what you would expect or want to see in atopic dermatitis. So this histology is again, low throughput. So we had this capability to measure electrical resistance through the tissue. So essentially, this instrument can put two electrodes, one on top and one at the bottom, so you can measure electrical resistance as a measure of the barrier of the tissue. And what was interesting is that as you go in the air liquid interface with time, you form the barrier. So the electrical resistance goes up, and those are the sort of bars in blue. And what happened with atopic dermatitis is that this barrier function doesn't form, so your tear levels are lower. And actually, this is a very HTS-friendly readout. You can do it in a 96-well plate, and it's fairly quick. So this was one of the assays that we used to then measure the effect of compounds. So IL-4 signals through a sort of a STAT-JAK pathway or JAK-STAT pathway. So what we did was to use JAK inhibitors to see if we could prevent the disease phenotype. So again, this is histology, H&E staining, normal, atopic dermatitis. You see the differences, the hyperproliferation on the epidermis. And then when we added the JAK inhibitors at the right timing, so we had 3 of them, we were able to correct the disease phenotype. We were interested in dexamethasone as a sort of immunosuppressor. And again, going back to the aspect of domain of validity, what are we really capturing in this particular model? Since we added IL-4, we didn't capture any of the T cell sort of polarization, any the sort of secretion of cytokines by T cells. So we expected that dexamethasone wouldn't work, and it didn't really work. So here's H&E staining. This is the different markers showing that the compounds were able to correct the phenotype. And then also I mentioned the tear, and what's interesting is that, so here you have the normal skin. This is atopic dermatitis, so reduction in tear. And then you see how the JAK inhibitors are able to correct the tear as well, and dexamethasone wasn't able to. So this was a sort of pharmacological proof of concept that we could use compounds to correct this disease phenotype. We went on to measure cytokines, and some of the cytokines that are relevant to the clinical sort of biomarkers of atopic dermatitis. So here are 3 of them: IL-1 alpha, TARC, and eotaxin. And what we did was to compare like a tissue that was only epidermis, a full thickness tissue, and a vascularized full thickness tissue. And what was really interesting is that for IL-1 alpha, really there was no difference, normal versus AD, and for the epidermis only. For the full thickness, when you added AD, so IL-1 alpha went up, and so it did TARC, and so it did eotaxin. And again, these 2, TARC and eotaxin, for the atopic dermatitis, it went down a little bit, but didn't go up, which is what you would expect for a clinical phenotype. So what this is telling us is that if you want to model atopic dermatitis in your skin, you need a full thickness skin. An epidermis only is not enough. You need to have the sort of fibroblast, an ECM layer underneath. And again, when you added the compounds in each one of these 3 cytokines, the levels were reduced to normal or even below. So it was a kind of a nice example that illustrates the question that I mentioned before: how much complexity you need in your tissue to model a disease. In this case, you need to have the dermis with the fibroblasts. In some of these tissues I mentioned, we had vasculature. So we actually look at the vasculature and what happened to the vasculature in an AD or atopic dermatitis phenotype. And what you can see here is when it's a normal tissue, you see a nice sort of vascularized bed. When you have atopic dermatitis, you kind of see, this is kind of the pattern in which the vessels were printed. So essentially, the AL4 prevented almost like the angiogenesis of the vessel. And this is quantified in these plots here. And then what happened with the JAK inhibitors, I shown that they can correct the disease phenotype by histology and tear, but they actually had a detrimental effect in the vasculature. And the JAK inhibitors actually reduce and really like sort of were not very good for the vascularized tissue. So in one way they corrected the phenotype, and on the other day, in another sort of way, they seem to be cytotoxic to vasculature. So even though vasculature didn't really help with the atopic dermatitis, it provided a way to sort of almost look at sort of side effects of compounds that otherwise would be corrective of the disease phenotype. So very quickly, again, I mentioned that some of the disease phenotypes work only in the full thickness skin, not in the epidermis. So this is just summarizing the data and really sort of highlighting that these 3D models are really more relevant than 2D models, because some of these cytokines, we really couldn't see them in 2D cultures of the keratinocyte fibroblasts and even in the epidermis. So this is one example where 3D really captures better disease phenotype. I'm going to move on quickly, 20 minutes, to the work that we've been doing for COVID-19, mostly on long tissue models. And a year ago, we were actually developing long tissue models for cancer, both metastatic and lung cancer models. And then when the COVID-19 pandemic came, we pivot these long tissues to study viral infection. And really what we're trying to do now is create a platform of tissues that can be used to study not only SARS-2, but any respiratory virus, sort of past, present, and future pandemic. So we're working on lung tissues. We sort of have some work going on kidney, intestine. We have neuronal models, and then endothelial and vasculature models. So we're trying to capture the sort of the tissues that from reading the literature seem to be affected by the virus. But the work that I'm going to be showing today is really work on the lung tissues. The strategy that we used was is shown in this slide, and again, it really follows the concept of like the concept of increased tissue complexity or progressive tissue complexity. The concept of developing these biofabrication techniques that are like versatile and modular, so you can actually choose what cells you want to include in your tissue and what sort of physiological features you want to include. And then we also, and I'm not going to show today, but we also started working in some microfluidic based platforms. And the strategy really, because of the urgency of the pandemic, we started working with some tissues that were commercially available by Biomarkers. You're probably familiar with the epi-airway and alveolar tissues. So we've been using those tissues, and at the same time, we've been developing our own tissues in a 96 well plate format, as well as the microfluidic So we sort of have tissues that cover different regions of the lung, the tracheobronchial, the small airway, and the alveolar regions of the lung. So the MATEC tissues they come in a transwell format, and again, the brain, the lung is really sort of in some ways very similar to skin. Nature is actually pretty redundant. You have an epithelial layer of cells that when you expose them to an air liquid interface, they differentiate into different type of epithelial cells. And then underneath, there's a sort of a tissue base made up of fibroblasts and vasculature. So the MATEC tissues, one of them, the epi-airways, the tracheobronchial, is essentially epithelial cells differentiated at the air liquid interface, and it includes ciliated cells, goblet cells, and basal cells, and that's shown here. And then the epialveolar tissue that they provide, it includes the alveolar epithelial cells, the T1s and T2s, fibroblasts, and then there's a layer of endothelial cells. So I'll show some data with these tissues. But then again, we were interested, this comes at one format. So we wanted to be able to have our own platform where we can have sort of the high throughput screening aspect of it in a 96 well plate, and also the ability to change the cells or include vasculature. So we've been doing a lot of work with small airway epithelial cells, and those are all primary cells, human primary cells. So this is sort of the basic protocol that we're using to manufacture these 96 well small airway lung tissues. Start with the cells. We culture them for a week in the particular media. Then we seed them in a 96 well transwell. And then you go for a week to let them make a nice coating. And then you go into air liquid interface for 3 weeks. And then again, a lot of the techniques that we use to validate these tissues are the techniques that I mentioned before: electrical resistance, immunofluorescent staining. And this is what I'm showing at this slide. Right now, we have a tissue that is only the epithelium, and we can see nice ciliated cells and with the alpha tubulin markers and the hairy cells. So these tissues differentiate quite well in 96 well plate. When you put them in the air liquid interface, you can see like the increase on the barrier function is measured by tear nicely with time. And then we've been doing a lot of imaging, 3D imaging, to not only look at the markers of the different type of cells in these epithelium, lung epitheliums, but also expression of the sort of SARS-2 receptors to see if they are expressed. And I must say that these tissues are actually very dynamic, depending on the week, depending on the media, the sort of the type of cells that you have, the expression of the ACE2 actually changes quite a bit. So when it comes down to a standardization, I must say that some of these tissues are actually, you need to be very careful and be very rigorous as to when do you measure what and what media you use. We actually spend a lot of time developing medias, developing conditions to grow the cells and differentiate the cells, and really trying to benchmark what we get to studies that we see in the literature. So the idea is that we will create these tissues, we'll infect them with the virus, and then whether it's by direct antigen staining in a high content screening assay or looking at secreted bios and measuring plaque forming units. We're very interested in secreted cytokines, and I'll show you some data with single cell sequencing, and then again, electrical resistance measurements. So this is just one example. This is the alveolar tissue infected with influenza. A lot of the work that we started doing at the beginning was with influenza because it was VSL2. So here is the mock, no virus. Then this is the alveolar tissue. So you get the markers for the different cell types in the tissue. No virus is a nuclear protein of the virus. And then as you increase the virus MOI, then you can see that you can start detecting the virus in the tissues with using imaging. And we've done, so that was one strain of influenza. We've done that now with actually two strains of influenza and SARS-2, and this is the tracheobronchial tissues, and this is the alveolar tissues. And what is really interesting is that the infectivity of the virus really depends on what tissue you're working with, and really on the virus, the timing, what time the virus peak, the infectivity peaks is very different between strains of influenza virus and between tissues. And then this column here is the SARS-2. And again, we think that this is actually a very useful model to just study the disease, study infectivity, study what we're planning to do is use these models to actually study the infectivity of the different variants that are now coming up of SARS-2, and really explore like what are the kinetics, what are the different sort of aspects of what cells get infected. As you can see, really the distribution of the infectivity, the SARS-2 is kind of patchy infectivity versus the influenza is like different. I mean, they use different receptors, so maybe that's expected. So this was imaging, and here I'm showing data looking at plaque forming units, and you can see again that the kinetics of the virus infectivity is different. This is for alveolar tissue. So these are the two strains of influenza, and this is the SARS-2. We were very interested in cytokines because it's sort of just from reading the news about the sort of COVID-19 cytokine storm. So we were, even though these tissues don't have any immune cells, we have not yet introduced immune cells. We're very interested in seeing, well, with the cells that we have, do we get any sort of cytokine secretions? And you actually do get cytokine secretion. Some of them are pretty relevant to the COVID-19 disease, like IL-6 and IP-10, have been sort of suggested as markers of disease progression for COVID-19. So this is actually influenza, and this is for SARS-2. And you can see again the differences between the different tissues. This is the tracheobronchial tissue. This is the alveolar tissue. So tracheobronchial seem to be more like sort of maybe not as many, not sort of the amplitude of the response is not as much. So you see here that the scale bar is different, but as you can see in the alveolar, the IP10, which is sort of a good marker of COVID-19 degree possession, really goes up after 6 days of infection. So it really sort of, again, going back to the aspect of can we use clinical biomarkers to benchmark our tissues in terms of clinical relevance. So we actually also been very interested in looking at these tissues again for the validation and standardization, looking at transcriptomic analysis. So we've done single-cell RNA-seq for these tissues. And again, I'm going to show data for both alveolar and tracheobronchial. So we did single-cell sequencing, and I mentioned that the alveolar had endothelial cells, fibroblasts, and then here you have the cluster of the different sort of epithelial cells. And the tracheobronchial, they don't have fibroblasts and endothelial cells. So these are the different type of cell differentiated epithelial cells in the tissue. So we've been able to sort of ID the different cell types in these tissues, as we should have by the imaging markers. And then we also look at sort of differences in the sort of gene expression. And then we've been looking at the different markers, especially looking at receptors. And here I'm showing data for the expression of ACE2, neuroglin, and TRAMPAS SS2. Those are sort of being suggested as as receptors for SARS-2. And you can see that ACE2, there's not a lot in the alveolar. Neuroglin, there's quite a bit in one type of cells, but not in the other ones. And TRAMPAS2 is actually the opposite, not much in this type of cells, but quite a bit on this type of other cells. And for the tracheobronchial, there's actually not a lot, and it's really across a different type of cells. So this type of analysis, I think it's very interesting because it really allows you to just, these are not infected tissues, these are just the normal tissues, and see where are the receptors, what cells express the receptors. And if you need both receptors, the number of cells that have both receptors is actually very small. So we still are going through these data, but it's turning out to be a very rich set of data. And then you can then infect these tissues and do the comparisons plus minus virus, and you see huge changes in the transcriptome. So this experiment was done in 48 hours. So here's the Mogno virus, here's the virus infected, and this is the alveolar, and this is the tracheobronchial. So huge sort of shifts in the populations. And you can have then look at what genes are the ones that change the most. And here you have the control, none without virus, and this is the virus. And what is very interesting is that within the infected tissues, you have infected and non-infected cells. So not all the cells get infected. And there's a set of genes that really at this time point, which was 48 hours, that this massive changes in gene expression, and as you can imagine, most of them are interferon pathway. And you can see both in the alveolar and the tracheobronchial tissues. This was done with influenza, and now we're doing it for SARS-2. And again, the idea is that we'll have these data sets with influenza, different strains, with SARS-2, and then hopefully other virus with time. And then what we're doing also is sort of doing different time points at 24, 48, 70, and sort of a week time point to see what the changes are a long time for gene expression. So this is kind of looking at the tissues, validating the tissues, establishing the clinical relevance of these tissues that we're making. And at the same time, we established a collaboration with Dr. Sarah Cherry at UPenn they set up a screening platform using a long cell line CALO3, and using high content imaging screen, they were able to screen for compounds that had anti-SARS-2 activity. And so we collaborated with her, and we took some of the compounds that came out of her screen, and we've been testing them in the sort of these tissue models. So this is just, again, some data with SARS-2 or no infection, and this is some of the compounds. So we see very nice activity with remdesivir, with interferons, a little bit with camostat. And then you can do the same exercise with influenza. So you see the virus here, and then you can see both the activity of the compounds preventing the viral infection. And here the green and the blue are the two tissues, the tracheobronchial and the alveolar, and we're going to generate the same data for SARS-2 as well. So just to give you a sense of how we go about doing the pharmacological validation of these tissues, and really establishing, benchmarking them for drug screening. So this is sort of what's next for us and what are we working on. So again, I mentioned like sort of the tissues that just had the epithelium, some tissues, the alveolar that had fibroblasts and endothelial cells. So for us, it's really going to the next level, sort of vasculature into the tissues, and really what happens to the vasculature if you infect the tissues. Do we see any evidence of maybe thrombosis, evidence of fibrosis? So we were interested in looking at that aspect, and also we're very interested in adding immune cells into the tissues. And then we're working, again, I'm not going to show any data, but we're working with sort of a microfluidic platforms to study the effect of flow, and whether you need flow to see some of both the markers, relevant markers of COVID-19, as well as pharmacological events, whether it makes a difference or not. So this is my last slide, and again, I sort of wanted to give you a couple of examples to see how we go about developing these 3D models. We do spend a lot of time sort of exploring different cell types to include in the model from different sources, from different companies, from different loads. So cell sourcing is a huge issue for us. Tissue assembly, it's also important, not because we have a bioprinter, we bioprint everything. So we use it when we need it. And then developing assays for validation, whether it's imaging, histology. Now with the single cell RNA-seq is a very powerful technique, and we're looking into some mass spectrometry techniques to see if we can look at sort of small molecules in the tissues. That's something that is quite relevant for us. And the whole aspect of implementing screens and what readouts they use. And you're going to be generating a lot of data, so you need to have a very good IT infrastructure to support all the storage of the imaging and the analysis and so on. So it's a tremendous group of people, dedicated people that have been working through the COVID-19 pandemic to develop these models and test the compounds. I'm really grateful and thankful to all of them. And I'm thankful to all of you for listening. Appreciate it.
Marc, that was a fantastic presentation. And those Star Trek members who are joining us right now know that there is a roundtable discussion to follow this presentation starting at 11. So I'd like to open the floor to all of those with us today that are not Star Trek members, so that they can ask their questions right now. Questions, anyone? Marc, I'd like to ask a question. So the data that you showed, it was really neat the way that you incorporated, you developed models with varying complexity for eczema, and then we're able to go back and answer your question of how much complexity do I need to answer the questions that I'm looking for. And I know that you said a future direction of the lab, an unmet need, in your opinion, is to start to add in the immune component. But I'm just curious, since you had those very compelling results already, if anyone in your group has gone back and added in some immune components to the full skin eczema model.
Yeah, we're just discussing doing it now. So we're very interested in sort of trying to it's the concept of divide and conquer, right? So you have different stages of the disease. Can you make models of the different stages, right? So the initial stage is an allergen that somehow interacts with your skin, and that triggers an immune response, right? So that's known to be driven by Langerhans cells, by dendritic cells. So one of the aspects that we're very interested is, can we include these type of cells in the skin to see if we can kind of address the sort of first stage of the disease, the sort of triggering aspect of the disease, right? And then another aspect would be, well, instead of using IL-4, why don't you guys use Th2 cells to induce the phenotype, sort of moving a step back, right? So that's an area that we're actually working with a collaborator, not for atopic dermatitis, but for psoriasis. And we have a collaboration where we take, in that case, for psoriasis, is Th17 and Th1 cells. So we're trying to use those cells, actual T cells, human T cells, to see if we can induce the phenotype. And what would be very interesting is really how different it is when you use the T cell versus using the cytokine, right? Because maybe there's this one cytokine driving it, but maybe it's a cocktail of cytokines, right? And how is that different if you if by using, let's say, Th2 cells, you get slightly different, your responses are different, right? So that to me would be sort of a next step that would be very interesting to explore.
Hey, Marc.
Yes.
I have a question.
Hey, Sita.
Very nice presentation. I was a little curious about the toxicity that you described with the JAK inhibitors on the vasculature of the skin.
Yes. Yeah.
I mean, that the atopic dermatitis, I mean, that I think JAK inhibitors are being explored very extensively. So I was wondering about the toxicity you saw on the vasculature. Did you see that only in the atopic dermatitis model or the normal skin as well?
Yeah, we're sort of exploring that more now.
Yeah. What kind of toxicity is that?
Yeah, we don't know yet. I think there's some evidence that JAK inhibitors are tricky from the clinical point of view.
Yeah.
And so whether there is some evidence that the JAK inhibitors can be anti-angiogenic, and the question is, well, how bad it is for, if you're going to use them for atopic dermatitis, how bad is it, right? Whether you can give them topically, or if you give them systemically, is that an issue, or what sort of toxic effect you see in clinic? But I think there's some papers saying that JAK inhibitors can be anti-angiogenic. So it wasn't totally unexpected.
But some of them are commercialized already. They are in.
Oh yeah. They're approved. Yeah. They're approved. Yeah. And they're being developed also for psoriasis. And look, I'm not a clinician, but I wonder if some of it will be sort of a matter of dosing and how you clinically prescribe them, right?
Yeah, in fact, one of them is being tried for clinical trials in combination with remdesivir, particularly
That'll be interesting too.
Yeah, that's why I was kind of interested in that. But that is for COVID though. But
yes. No, I got it.
Yeah, that's not for atopic dermatitis. Okay. Yeah.
I saw that you have a question in the chat, Sita, about 2D versus 3D.
Yeah.
And this is something that it's in the to-do list.
Yeah.
So looking at not only infectivity, but the effect of the compounds with your models in a 2D submerged sort of standard, let's call it standard HCS protocol. So that's something that we're very interested in looking
Yeah, I
at.
think that'll be very useful to the general public because whenever they are using 2D, cultured cells versus whether they need to go to a complex model like this. That's the reason I asked.
No, it's always the question, right? Why do you want to put more work on yourself if using primary cells in 2D give you the same answer, right? For this particular effect, right? If what you're interested in is like viral infection, I mean, from virus to CALU3, it seems that it's much better. And then the question is from CALU3 to primary sort of epithelial cells, how much more you gain, and then from the primary epithelial cells submerge into a 3D lung model, how much more you gain. I mean, ballpark, I would say half of the compounds that were active in a CALU3 infectious assay, maybe half of them reproduce in the tissue models.
I see. That's interesting.
So I mean, I think part of the issue is that, we ACE2, the Trump SSS2. So what is really the receptors and what are the receptor requirement is not that clear either. And the mechanism of entry, and it just sometimes what you find is just doesn't reproduce in these like tissue models.
Yeah, there's a discussion going on in a separate venue about how do you incorporate in this model the effect of aging. It seems to be that the infectivity is very high in older people and who are vulnerable. So how would you engineer?
Yeah, that's the one aspect of the discussion, right? If you can get good reproducibility with one lot of cells, and then you get cells from patients of different age, patients with asthma or patients with diabetes, you get cells from these different populations, and then you do the same exercise. First of all, are you going to see different infectivity? Second of all, when you do single cell sequencing, are you going to see different responses? If you make cytokines, are you going to see different cytokines, right? And then would the compound work the same or not? So it's a very interesting sort of exercise, and I think having good sources of cells that are well sort of established in terms of who the patient was, I think those are very important as well. I mean, right now, primary cells is our sort of favorite, but you really have to worry about lot. And then you can sort of go into a generic sort of tissue with iPS-derived cells, and that's an appealing approach too, because technically you have unlimited sources of cells.
Right.
But again, you have to worry about clone-to-clone variability, because there's been some clone variability when it comes to iPSs.
But that's true with primary cell sources too, right? So.
Oh yeah. I mean, I would say most of our work is really validate sources of primary cells. And then finding the right media to grow them and put all the cells together.
Yeah.
Marc, Kenneth Miller, who's with us, wrote a very interesting question in the chat. If you don't see it, I'm happy to read it.
Yeah, I don't see it.
I don't see it either. Yeah.
Okay, so it says, really cool stuff. Regarding the lung models, how do you control not only variability between experiments from a single donor due to differentiation, but donor to donor? How do you settle on a representative donor that you can draw conclusions that are widely applicable? Very good question.
It is a very good question. And sometimes the answer is actually very practical. Sometimes we test 4 or 5 batches of lots of cells and see which ones give good differentiation, good tier. Then if we have different lots that give us the same result, we essentially purchase the lots with more vials. Because again, I mean, you want to address the technical reproducibility issue. You want to know that with cells from one lot, you can have like a 96 well plate with good CVs, with good infectivity, reproducible infectivity. And it's really, once you address that reproducibility, then you might go to the next lot and say, do I get the same in the same conditions or not? To be honest, we haven't done this very systematically because it's a lot of work, but it's to the point of this discussion and this series, it is really what is needed, because if you can do that, and then you can like sort of like, okay, this patient, what was this patient, age, race. So then you can correlate your sort of differences of similarities in your tissues to the background of the cells. That becomes very interesting. But we haven't been able to do it very systematically. I think within this year, we essentially developing the protocols. And I think now we are at a point that we can go to a different lot and say, okay, if I use the same conditions, what do I get? Very good question. A lot of work. And it's a kind of ungrateful work, right? Because it's really very ground sort of comparing, sort of establishing what the variability is, but it is very critical work. I agree.
So probably you're going to have precision medicine generate tissues for each individual. That's what probably you're going to.
Yeah, I mean, if we're going to get to that point and we want to do that, we need to do the systematic work of comparing.
Yeah.
But first, you need to have the technical reproducibility.
Right.
Do we have questions from any other non-Star Trek members? The excellent discussion so far. Okay. Well, if there are no other questions, then we'll go ahead and begin the roundtable discussion with the Star Trek